connect that. Where are we here? Day 2 by, here we go. Okay. All right. So yesterday we covered the topic of bistability, and there are a couple of things we mentioned. Maybe I can summarize them here. Number one, bistability means two states. Okay. But there is always an unstable state. That's how you can get from one to the other. So there's actually three states, but two of them are stable. Two examples of bistability. One was a simple positive feedback. I can type. All right, that was one. That was just two reactions. Two reactions, and the intermediate species had a positive feedback on the first reaction. The second one was a toggle switch. Okay. And the other important thing, which I guess I'll show you by picture instead, was this idea of hysteresis. So, and I used a model from John Tyson's paper that also had positive feedback. And this one had an input signal, S. And what you can do is you can vary the input signal, and that actually changes the behavior of the system. And at low signal, there's only one state, so it's not bistable. So if you look at this picture, let me make it bigger. If you look at this picture, so these are all the steady states. The R was the output. These are all the steady states as you vary S, okay, the signal. And you notice bistability only exists in a sort of narrow range here between about 0.8-ish and 1.7 or so. Outside that range, it's not bistable. In fact, this is something I didn't mention yesterday, I don't think. So down here, this lower bit is only one state. Okay. Up here is also only one state. Right? These are not bistable in these areas. But if you have the signal S within a certain band, the system does show bistability. And this is very typical of systems in general, right? They change behavior as you change a parameter, in this case, the input S. But in this case, we get a special situation where you get this bistability appearing. And these two arms here, the pure blue one and the pure red one, represent the two stable regions of this system. And one of them is a high state, and the other is a low state. In between them, there's this green line, which roughly marks where the unstable state is. Okay, so there's an unstable state between the two. And, you can jump back and forth. I showed you some simulations where you can jump back and forth between these. And, but the jumps, if you increase the signal, the jump to the high state occurs at a different point where if you were to decrease the signal from the high state, it jumps at a different point to the low state. So this phenomenon is called hysteresis. Very common, found all over the place. Okay. So now this leads me on to oscillatory circuits. So you may think this is a bit of a jump. Is there any relationship between bistability and oscillatory circuits? Turns out there is a really deep relationship. So let's talk about oscillators. So what I've got here is a picture of this nodding duck. I it's called a drinking duck. So I teach this topic in a class in January, March time. And I bring in some demos, and then I bring in my little duck into the class. Along with a couple of other things, I have also some electronic circuits which oscillate. But this duck also oscillates. Basically, it nods up and down, right? And there's water in that cup, as you can see. I think you can see there's water in the cup, preferably slightly warm. But the duck's beak serves as an evaporation point. And so when you dip the duck into the water, his beak gets wet. And when he pops out of the water, the water starts to evaporate from his beak. And when water evaporates, that requires energy, and that energy comes from the cooling of his beak. And the cooling of his beak cools the air in that glass tube, which reduces the pressure, which means that this alcohol here, I think it's alcohol, gets sucked up the tube. And when the alcohol gets sucked up the tube, he tips over because he's now top heavy, and he goes back into the into the glass beaker, and because and the water then will cool down his beak. That will increase the temperature, pressure, and the alcohol will flow back. Actually, there's a little hole here, so when it tips, when it tips up, um, it exposes a hole, and the alcohol just pours back down. That's what actually happens. But anyway, his beak is re-wetted. The thing falls back down, and the evaporation starts again. And so he'll go on nodding like this for for a long time. Um, Quite fun to watch, I guess. But he's an example, or she is an example of an oscillator. And there are all sorts of oscillators. There's electronic oscillators, as I mentioned, but there are also biological oscillators. And you might think, yeah, well, there's no relationship between any of these oscillators, surely. Yeah, well, it turns out they all work in the same principle, even our little nodding duck. So this is what we're going to talk about. And before somebody says, yeah, but oscillators great, but what's it got to do with biology? Well, oscillators in biology are all over the place. In fact, I think there are far more than we realize, especially inside cells. We have circadian rhythms, of course. We have the heartbeat, cell cycle, P41 DNA damage repair is an oscillatory system. But we also have more exotic things like cell division in prokaryotes, which involves an oscillator. It uses the oscillator to find the center of the cell so it can then divide. And oscillations have been seen quite often in signaling networks. I mean, nobody knows what they're doing there, of course. I mean, these ones we see here are pretty obvious, although why P41 DNA repairing DNA requires an oscillator, I don't think people quite understand. Although one hypothesis is that it could act as some kind of counter, and if the count goes too high, the cell then decides, yeah, the DNA is too damaged, I can't do anything about it, and it goes self-destructs. So maybe that's what it is, but we don't really know. Okay, so lots of oscillators in biology, worthy of understanding. All right, so this is a little system we had yesterday at the beginning. This is a little bistable system, innocent looking thing, just 2 steps, and a negative feedback from S2 back to this reaction here, positive feedback, okay? Positive feedback. I've numbered this in this way because if I add one more step, right, this oscillates. Okay, so if I go from this, I'll make sure that the S1 and X1 are fixed on this model. We discover it's bistable. If I now add an extra step, relax this now so that it floats, but fix this one, X0, this oscillates. I mean, what have we done to make this oscillate? I mean, all we did was added a simple step. This is just a simple mass action step. There's nothing special about it. The only thing that's special is this positive feedback. Okay, so that's an interesting problem. In fact, just to convince you, no, here it is actually. Why don't we go ahead and run it? This is the model. I'll take it on this one. Of course, I'm going to have to now redo this. Let's just do that first. I suppose I should have done that before. On the other hand, I could do this, new, paste, run. Invalid syntax. Oh, yeah, of course. Almost as fast as starting up
Colab.
Starting up Colab, except we got the wrong time there. There you go. All right, let's go back to Colab. Should be ready. Almost, yeah, should be ready now, hopefully. So I'll import tellurium. Just, I forgot. Yeah, of course, I forgot that. Yes, yes, yes. Good. And then I'll run tellurium. Now it's happy. Oh, almost happy. Come on. There you go. All right. So now I'll create a new cell. Add the model. Let's do a simulation. Simulate. Let's go up to 500. Let's have a lot of points so we don't get any jaggies. And then plot it, and you'll see that it oscillates. Okay, oscillates. So all I've got here is first order. In fact, this is a constant injection of mass into the system because K0 is constant, X0 is fixed. So the product is a constant. So we have a constant injection. Then I have this special rate law here, which provides a basal rate plus this positive feedback component. Then I have a simple, mass action linear rate law there. This oscillates. Right? Amazing. Okay. So let's see if we can figure out why. There it is in all its glory. One thing to notice about this oscillation is that you'll notice there seem to be multiple timescales here. There seems to be a fast timescale where this spike is changing quite rapidly. Then there's a slow, much slower movement. Okay. And this is very characteristic of an oscillator which depends on positive feedback. In fact, this kind of oscillator has a particular name. It's called a relaxation oscillator. They're very common in electronics. What's good about them is they only require two variables, S1 and S2, and they can produce quite varied waveforms. So those of you who've done any electronics will perhaps have come across signal generators. These are sort of boxes on your workbench that produce various signals like sine waves, square waves, sawtooth waves, and so on. Almost always those are using relaxation oscillators. Okay. And in fact, a lot of these processes I mentioned here are also relaxation oscillators. The heartbeat is a relaxation oscillator. The cell cycle is a relaxation oscillator. Circadian rhythms, I don't know. P41 DNA damage is also a relaxation oscillator. So they're actually quite common. I mean, we don't know for certain why cells use them, but definitely in man-made devices, one reason is because it requires fewer components, and the second reason is you can have a huge variety of profiles on the waveforms. Okay, so what's going on here? Well, there's actually a hidden negative feedback. It's rather difficult to see in this model because it's not explicit, but there is actually a hidden negative feedback. So let me see if we can figure this out. So there's definitely a positive feedback, and that's written into the equation, right? There's no doubt about that. So let's imagine that positive feedback increases S2. All right. Now, if S2 increases, this will stimulate the positive feedback even more, which increases this reaction rate, which means that S1 gets consumed more. So in effect, S2 is having a negative effect on S1. Okay. And S1 itself, of course, has a positive effect on the reaction rate. So the net is that there's an overall negative feedback between S2 and the reaction. Right? You may think that rather odd, but that's actually what's happening. So I'll say it again. If S2 goes up, it stimulates production of more S2. This rate goes up. If this rate goes up, S1 goes down because it gets consumed faster. So in effect, S2 is causing S1 to decrease, right? It's acting negatively. And because S1 has a positive effect on the reaction itself, right? Remember the basal rate? There's a net negative feedback. And it's that combination of a negative feedback and a positive feedback that results in this relaxation oscillator. It's conceptually it's a more of a complicated oscillator than another kind. There are really only two kinds of oscillators: relaxation oscillators and feedback oscillators. The feedback oscillator is relatively simple to understand. We'll come to that shortly. The relaxation oscillator, though, is fairly common, but it's more difficult to understand. Okay. Oh, right. I was going to show you something. Yeah, let's show you this. So, one of the things with Python is that simulations are generally static, right? I get curves like this. Okay. So I hit the run button and I see, a set of curves. Change the parameter, hit another run button again, see another static image. Wouldn't it be nice if we had a more interactive simulator? And we have a tool. It's not yet released. It's sort of in beta, which I shall show you. Here it is. Runs on Windows and the Mac. Called it Iridium because that's also an element next to antimony and tellurium. But it's specifically an interactive simulator, right? It's for interactive work. So it sort of complements the Python side, right? And what I've got here is we've got a window here where I can drop in antimony code, right? An antimony model, or you can load in SVML. Either will work. And this model I've got here, if you can see it, maybe if I'll zoom up a bit here, that's actually the relaxation oscillator. Okay, so there's the positive feedback, there's the reaction in front, there's the reaction behind, here are the parameters. Okay. I can run a simulation by just hitting the simulate button and I get this plot here. That works quite nicely. But what's nice is I can bring up a bunch of sliders. So if I press this button, I get a bunch of sliders, although it doesn't bring up any initially, but I can say make all sliders. So this, what it does is it attaches a slider to every parameter in the system. Okay. And so now I'm going to cut off auto scale. Otherwise it'll be a bit annoying. And I'll take that to 5. There you go. Keep the X auto scale on. And each of these parameters corresponds to a different parameter. For example, the Hill coefficient, N, which determines the strength of the positive feedback, I can mess with, right? So I can increase this, and of course you see that changing. But if I bring it down low enough, the oscillations disappear. All right. So the strength of the positive feedback is definitely important here, right? I can bring it back up and off and up it goes, right? But I can also mess with the other ones too. These also have different effects. If K0 is too high, i.e. the injection of mass into the system, it also stops the oscillations. That changes things. The Vmax, that changes things, and this other one changes things, okay? This is the point I made about in the bistable system where I was changing the input signal and I was moving from non-bistability to bistability and then out of bistability. This is the same thing where I can move from oscillations to not oscillations, right, and back again. All right. So this is the one thing I use this tool for. I've used it quite a bit for that. Is I have a model and I am suspicious that it probably can oscillate. You can start messing with these to try to make it oscillate manually. So you just fiddle with the parameters until you find it does oscillate, and it usually quite works. Because the different way to do it is to write some kind of optimizer in Python that looks for the parameter settings that give you oscillation. Right, but this is probably more fun. All right. So as I said, this is in beta, this tool. There's a bunch of other stuff it can do as well, like steady states and real-time simulation and things like that. It's in beta. I got a couple of groups who are trying it out. They give me feedback, but I'll give you the link if you're interested in trying this out. And if you do find bugs, let us know, okay? Yeah, okay. So as I said, the software is still in beta, but there's the link if you want to try it out. We've also decided to move it to the web. So there's no installation. You don't have to do any installation, and it'll work on any platform, including your tablet or whatever, or your phone, although I don't recommend the phone. It's a bit too small. But we have a prototype of some of it running now already on the web. Hopefully that'll come out at some point. Okay. All right. That's the screenshot. Okay, so what's actually happening with this oscillator? How is it managing to oscillate? And what's the importance of the negative feedback? So if you remember, we had this hysteresis plot for the bistable system. Remember, it was that S shape. If I were to fix S1, right, and just keep this portion of the network, and I looked at the steady state level of S2 as a function of S1, which is now fixed, so I can change it, you'll discover there's a hysteresis in the system, and that's the bistability. So at low S1, it's not bistable. At high S1, it's not bistable, but in the middle, it is bistable. It is bistable. And what the negative feedback does is effectively moves S1 up and down, up and down. That's effectively what it does. And so as S moves up, for example, let's say when you start up the simulation and the positive feedback is weak, S1 will start to rise, okay? And as it rises, it moves up this line. But when it gets to this point, it's got no option but to flip to the high state, okay? And in the high state, because as it moves up, because the rate goes up and the positive feedback becomes more and more pronounced, and it flips into the high state. And in the high state, there's a very high reaction rate because it's now flipped to a very high level of S2, which is really forcing the positive feedback. Because it's the high level of the rate is now at a high level, it starts to deplete S1, and so S1 starts to creep back down again. As it creeps back down again, and because of the hysteresis, it creeps back down on this line, it eventually gets to this little cliff face and drops off that down to the lower state. And in the lower state, the rate is low. And so S1 can start to climb again. And of course, you can imagine it does this again and again and again. And this is why we get the oscillations. Okay. The negative feedback here is crucial because it lets this thing move up and down like this. So that's essentially how it works. And I got a sort of diagram here showing it moving back and forth, back and forth. All the relaxation oscillators do this. Heartbeat, cell cycle, P41, DNA damage repair, they all basically do this. Okay. And in some sense, I guess, if you can get the hysteresis to work, it's not too difficult then to add some kind of negative feedback to make it flip back and forth. Okay. Now, this looks a bit complicated. All right. Turns out you can turn this into a very simple motif, which basically points out the core idea of this relaxation oscillator. Okay. So in this motif, I have two species, S1 and S2. S2 has a positive feedback on itself, meaning S2 makes more of itself. The more S2 there is, the more S2 there will be. But then it also has a negative feedback, right, with a bit of a delay on S1. You need the delay, all right? Bit of a delay on S1 that goes back to clamp back down on the S2. So you get this to and fro between the two all the time, okay? And as I say here, relaxation oscillator has two parts, a threshold device, right, which is the bistable system, and a negative feedback loop. And they all work on this principle. Now, it turns out this is sort of a general motif. We actually call this a design pattern. In fact, we have an entire paper published a couple of months ago with Steve Andrews and Steve Wiley on design patterns. And it describes these basic patterns you find in biology. This is the core principle as to how this oscillator works, but how you implement it is another matter. And in fact, this is from an old paper I had back in 2003. I can't remember when I published this, perhaps 2005, where you'll see this. I call it a stylized form then, but now I call it a design pattern. These three, and actually this one as well. Actually, all of the first five are essentially the same. These two, I've just put the negative feedback in a different place, but it's basically the same. You see all these different implementations. So there are many ways to implement this pattern, right? But at the core of it, they're the same thing. They're doing exactly the same thing. Okay. This is a different pattern, but actually ultimately it still works on the same principle as a relaxation oscillator. All right. So John Tyson in his paper, he also classified these as substrate depletion, activator inhibitor, substrate depletion toggle. Okay, he had some degree of classification as well. These are all oscillators. All right. I suspect there's probably an infinite number of ways. Well, that's a bit crazy, I guess, but there are a massive number of ways to implement oscillators in biology. Huge. Okay. Now it's pretty easy to make, well, when I say easy, it takes a lot of experimental effort, but conceptually, it's pretty easy to make a relaxation oscillator in vivo using genetic systems. So basically, you can start off with a gene with a positive feedback and then add on another gene which provides the negative feedback. Okay. So you have a bistable switch here connected to a negative feedback there. This thing will oscillate. Okay. Okay, I don't. All right, I'll show you some real examples of this later on. So this is the basis of this kind of oscillator. It's called a relaxation oscillator, very common. And if you dig into it enough, you can sort of understand how it works. Okay. Now you can also perhaps understand why you get these different time scales, right? The positive feedback, once it starts increasing, can be very rapid, right? But the change in S1 can be generally slow. Let's see if I got that right. Did I get that right? So S2 is the positive. Yeah. So S2 has a very rapid change, right? Very rapid, very sharp spike. And then S1 is a bit slower. Because as S1 is slower, S2 is a little bit calmer as well. But once it starts going, there's nothing to stop it. All right. So it's sort of almost like an explosive effect. And it doesn't explode forever. It doesn't go up to infinity because it's limited by the system itself. There are physical limits on the system. All right. Okay. What's the other kind of oscillator? The other kind of oscillator is a much more simpler one to understand. It's called the negative feedback oscillator. It's also called the phase shift oscillator. And it's really simple. It just has a single negative feedback. So you have a long line of reactions, and it has to be at least 3 reactions, right? If you have only 2, it won't work. If you have 1, it won't work. You have to have at least 3. And from the third species, you need to have a negative feedback going from there back to the first step. If you have that, and you have a strong negative feedback on this step here, you will get oscillations. All right. So this goes back to what's so special about negative feedback. We haven't talked much about negative feedback, so I'm going to say a few things about negative feedback. All right. Here's a very simple model that shows negative feedback. Just 2 steps, negative feedback back there, right? Pretty simple. So what I'm going to do here, these 2 reactions are V1 and V2, and I'm going to plot V1 and V2 versus the level of S1. Okay, so S1 is on the x-axis, V1 and V2 on the y-axis. All right. And all I'm plotting here are literally the rates. This is not a simulation. I'm just plotting the rates. I'm going to make the second step V2 just a simple linear rate law. All right. So the rate is proportional to S1, and that's why I get straight lines. Now, if I change the rate constant on this second reaction, I can get different slopes. So I can move the slope of that second line up and down as much as I like. The negative feedback, I'm going to use an inverted sigmoid, basically an inverted Hill equation, effectively what it is, and I get this inverted S shape. Okay. Now, this line here in between the 1 and the 2, I can make as steep as I want. All right. I've made it moderately steep here. But you can imagine that if I were to perturb the second step by changing the rate constant, I've effectively, as I said, changed the slope of this line, right? And when I change the slope of the line, this intersection point, remember we talked about these intersection points? These intersection points are where the 2 rates are equal and mark the steady state. So if I take V2 as 0.1, right, the rate constant is 0.1, let's say, it intersects here, and therefore the steady state level for S1 for the system will be about 1.9. If I move the slope up, the concentration decreases, and I get a steady state level about 1.5. Okay. That makes sense, actually. If I were to increase the rate constant here, this rate goes up, therefore S1 falls, right? So S1 moves from right to left. That makes logical sense, right? But notice that the steeper this line, the less S1 will move. Okay, and if it's almost vertical, if it were almost vertical, at this point here, S1 would barely move if I changed the rate constant here. And this is an example of the homeostatic properties of negative feedback. Negative feedback resists change, right? So if you start changing this consumption rate and the negative feedback is working, you won't see much change in S1, right? That's an example of homeostasis through negative feedback. That's one of its most basic behaviors, right, in negative feedback. Now, and that's a good thing, right? Everybody likes homeostasis, all right? I want some level of predictability, and that's what homeostasis gives me. But you can have too much of a good thing. Like, let's wrap negative feedback around a much bigger network. That'll give us lots of homeostasis. Well, not so fast, because if you try to wrap it around a pathway that's too long, it'll break into oscillations. Okay, so negative feedback is a two-pronged issue. Too much of it will give you oscillations, which sometimes can be a bad thing, sometimes a good thing. All right, but if you have too little oscillation, you get homeostasis, which is nice. But if you want stronger homeostasis, you have to strengthen negative feedback, but then you might end up oscillating. So there's this fine line between getting homeostasis and ending up with oscillations, right? And this is a really simple system. Oops, sorry. I've built, right? Let me grab this. Oops. Let me go to here. Let me create a new cell. There you go. So this system has 1, 2, 3, 4, 5 steps. Linear chain. I've put some, I don't think this is really necessary, but I put some Michaelis-Menten-like equations here. That's not so important, but the negative feedback is here, okay? So the negative feedback is in S4 to the power of H, all right? And the H determines, the Hill coefficient determines how strong the feedback is. So let's
Herbert,
dial that
a quick
down.
question. You've been using V's instead of K's.
Yeah, that's
just
yeah.
for convention or something. Yeah.
No, it's my error.
Oh, okay.
I realized that when I was talking. Those should be K. Those should be
Okay, sure.
K's. Okay. Yeah, that's my bad. There you go.
I mean, they're arbitrary names. K is just sort of the
Yeah.
traditional way of using these things. But yeah.
These V's are real though. Okay. These V's are real. I guess I was just trying to label these lines as the second reaction rate. Okay. But if we just use K2, that basically means the rate constant in the second reaction. And that rate law, let me just put it here. You can see that rate law V2, I'm going to make it like this. It's going to be just a simple linear relationship, right? So as I change S1, V2 changes linearly, right? And I can change the slope of that line just by changing K2, all right? That's basically it. And I'm using that changing K2 as a perturbation. It's a way I can perturb the system and see what happens. That's basically what I'm doing. All right. Yeah. No, good. Please ask questions. So what I've done here, I've got a much longer negative feedback loop, right? Much longer one. And I've got the Hill coefficient down here. Sometimes it's called N, sometimes H. but I've dialed it down to 1. So there's really very weak feedback. And if I run this, if I'm lucky, I shouldn't get anything. There you go. Stable as a rock, right? It's stable anyway. I mean, it's pretty steady. I could do a sensitivity analysis on this, and I would see that the system is quite resistant to changes. I could perturb, say, this reaction here. In fact, I should call that Vmax as
I don't
well.
know if you've defined what a sensitivity analysis is.
No, I haven't.
Okay.
Let me explain in terms of operations. So I'm at steady state. Let's do this, shall we? Let's go up to 100. Make sure we're at steady state. So everything is settled down. There we go. We're at steady state, I think. And we've got S1, S1, S4, S2, and S3. And I can ask the question, in fact, let's do it in an event. In fact, I'm going to go up to 200. And then I'm going to say if time is greater than 100, I'm going to perturb the Vmax on this last step, okay? All right. So Vmax times, let's say 1.5. So there's my disturbance, okay? So I'll run this, and there you can see an effect, right? Everything plunges quite a bit, actually. I've increased it by 50%, but I'm seeing fairly well also 50% drop in S4. So it's very sensitive to changes in Vmax. And that's basically what a sensitivity analysis is, okay? You're changing something and then you're seeing, well, what effect does it have? In this case, on S4, at least pretty dramatic. Well, let's try upping this Hill coefficient. Let's try strengthening the feedback. See what happens. Aha, look at this. Much better, I would say. Yeah. Well, not much better, but better, right? So here's the steady state. I've reached the steady state at about 100, right? Then I make my perturbation, the same perturbation. Now well, actually, it's still dropping actually quite a bit as well, though, isn't it? Yeah, okay. Although perhaps not as much. Let me see if I increase this even more. Okay, this is better. Yeah, this is better. Although I'm losing I need some more points here because it's getting a bit jagged. So let's do that. So now I've increased the strength of the feedback even more. And here I make that perturbation. It was at about 3.2 and it drops to about 2.8. It's much less than 50%. So you can see that the negative feedback, and I never tried this before, right? So I'm hoping that the universe is consistent and behaving properly. It actually resulted in reducing the sensitivity of the system to perturbations, i.e., it's behaving homeostatically. Now, you will notice, however, there is a penalty. I'm getting this, what's called ringing. So in engineering, this is called ringing, where you get a little damped oscillation when you make the perturbation, right? So you see here little few wiggles before it settles down. That's called ringing. Usually a bad thing in engineering. But if I keep on increasing the Hill coefficient, the ringing starts to increase and increase in amplitude, and it starts to last longer. In fact, there is a known threshold for the Hill coefficient at around 8 or 9, where the ringing actually becomes sustained. There, you see it there? Becomes sustained. Let's go to 9. And it just oscillates, okay? So if you're an engineer and you want to avoid oscillations, you want to make sure that the strength of the feedback is not too high. So you want to throttle back the strength of the feedback. And a lot of engineering is about that because negative feedbacks appear in a lot of places even without, you realizing it. And engineers are always grappling with that to try to keep their systems stable. And they have all sorts of tricks to try to pull back from this precipice, right, where you suddenly start oscillating. Now, in some cases, like if you're building a signal generator, you really do want oscillations, right? But if you're building a bridge or a rocket, you don't want oscillations, okay? And up here in Washington State, we have a very famous case of that, the Tacoma Bridge. If you look that up, was it in the 20s or 30s? It basically started oscillating violently and basically tore itself apart. So, engineers showed great efforts to avoid this kind of thing. Okay, so that's the little model there. Let's, oops. So as I said, it's called a negative feedback oscillator or a phase shift oscillator. And the reason it's called a phase shift oscillator is because not only do you need a strong negative feedback, but you also need delays. And the delays can be quantified in terms of how much they phase shift any perturbation that travels through the system. And as long as the phase shift is high enough, you will get oscillations. In fact, the phase shift has to be at least 180 degrees between the front and the back. If that's the case, it basically inverts the negative feedback into a positive feedback. That's essentially what's happening. So the negative feedback is meant to stabilize. In other words, if S4 goes up, the negative feedback is supposed to bring it back down, right? It's supposed to do the opposite. But if the phase shift is out by at least 180 degrees, the opposite happens. So when S1 goes up, when S4 goes up, the negative feedback now does the opposite because all the phases are out and it starts to increase it instead. And that's actually the source of the oscillations. And actually, maybe I can show you if I go to phase shift oscillator electronics, right? If I go there and go to images, you'll see lots of images of phase shift oscillators, and they all sort of look the same. Let's look at this one, shall we? Can I get this one up? Here it is. Can you see this? Let me zoom it in a bit. Yes, go away, go away. They look like this. And you may think this is nothing like the biochemical network you just showed us. So actually, it's very, very similar. There's the negative feedback unit, which in this case is a transistor. And the three capacitors are the three species in the network, and they act as the phase shifters. All right. So the signal goes all the way through the capacitors and into the base of the transistor, and that's the loop. Okay, so it's actually exactly the same thing as our biochemical oscillator. And all phase shift oscillators are like this. They have some place where you can enable the oscillation, the feedback. And that feedback has to be fairly strong, as you saw. Of course, the transistor is an amplifier, so that provides a nice, strong negative feedback. And then you have to have three phase shifters to get you up to at least 180 degrees. That's why you can't get an oscillator if you only have two species, because you can't get enough of a phase shift. All right. It turns out that each species here can contribute 90 degrees. With only two, you can only get up to 180. You can't actually hit it. So you have to have an additional one, which will take you up to a maximum of 270 degrees. And that'll ensure that there's at least one place where you can get exactly 180. And that's the thing that then destabilizes the system. Okay. So I don't know how well that was explained, but that's why these things oscillate. What you'll often hear people say, yes, the reason you get feedback here is because of the delays, but the phrase delay is very vague. What do you mean by delays? Because I know that with just two species, it doesn't work, but there are still delays. Then they say, well, not enough delay. Yeah, what do you mean by not enough delay? The real answer is to do the phase shifts that you get across the system. Okay. Let's see if we have time to do this exercise. But
we have about half hour total left.
Yeah, we probably don't have enough time then. So let's just carry on, okay? Because I want to do another project instead, okay? Here's another variant on the feedback oscillator. It's called a ring oscillator. To make a ring oscillator, you just need to have an odd number of inverters in a loop, in a ring, and it'll oscillate. It's basically the same as a feedback oscillator. May not look it, but it is. And just to show you, this is not a crazy idea, but you can actually make a model of a ring oscillator using 3 genes. Each gene represses the next one, and the last one represses the first one. If you do that, this will oscillate, all right? And I've got the model here. These are the 3 genetic units. The second reaction in each one is just the degradation step. The actual gene is P1, protein P1, protein P2, protein P3, and there's the negative feedback. Okay. Note that you may be asking, why do I need an odd number? Because with an odd number, with 2 negatives turn into a positive, and a positive times a negative is a negative, so it's net negative. If I had an even number there, it would just be a positive cycle. That I don't want that. I want a negative feedback net. Because not only that, back in 2000, somebody said, yeah, hey, why don't we build one? And so they did. They built one in E. coli. This is the famous repressolator. Some of you may have come across this back in 2000. They actually got 3 genes together. They put in 3 repressors. They had a reporter on the GFP. They built it. It took a while to get all the promoter strengths right in order to get sufficient amplification. It took a while, but they did it in the end, and the thing oscillates. Okay, this is a very famous synthetic biology paper. It's 2 that came out at roughly the same time. One was a bistable circuit, and the other one was an oscillator circuit. Okay. I could mention there are lots of people have built lots of other oscillators since. Here's one from Jeff Hasty. He built a feedback oscillator using LAC-I. You may be thinking there's only one gene here, so how can you get oscillations? You said we had to have at least 3 steps. Well, of course, between the gene and the transcription factor, there's a lot of stuff like ribosomes, RNA polymerase, and so on. And they provide enough steps to give you the delay, and so it oscillates. He also did a relaxation oscillator. Here's the positive feedback, right? And then the negative feedback, it comes all the way around the green, back to the green, back to the red. Okay, so that's a relaxation oscillator, combining a positive feedback with a negative feedback. That's the same again. He's got a bunch of movies, which are nice to watch, showing these things, E. coli blinking on and off.
Does the fact that it needs mRNA to oscillate mean that if you want your model of the system to oscillate, you need to include the mRNA as a step?
So yes, yeah. If you try to model this with just one reaction, it won't oscillate.
Right.
So you have to put the mRNA in, you have to put the ribosome in, and you actually need one more something, and it'll oscillate. But there are a lot of steps between this gene and repression, right? A lot of biochemical steps, and that
could you sort of fake it with a delay, something, or like an event or something?
I don't know. That's too fakery.
Too hacky, yeah.
Yeah.
Okay.
Yeah, too much faking there, I think. The other one I can mention is this nice one here. This is a synthetic mammalian oscillator. It's from a German group. This is a lot more complicated, but it produced some nice oscillations. This is also a relaxation oscillator. This is the circuit, and it looks very complicated, but actually, let me see. Okay, let me see if I can show you the bits. So this bit here in the middle is actually the positive feedback, right? If you take the gene here, TTA, this makes some mRNA, which makes the transcription factor, which then activates more production of TTA. So that's the positive feedback, right? From the positive feedback, we go all the way around this loop, right? All the way back to here to actually inhibit the positive feedback. So there's a combined positive feedback and a negative feedback. We have a whole reporter system up here. That's just the output, okay, with GFP. And this thing oscillates. This is a very nice example. It's pretty sophisticated, using lots of tricks to make this work, but that was a nice example. And there are lots of natural oscillators as well. I've got some papers here that if you're interested to look at. One of the famous one is the NF kappa B oscillator. In first oscillation, I think, detected in the signaling network, I think. There are quite a few oscillators in metabolism, in yeast metabolism especially. Nobody knows if those are useful to the organism or not, but you can make them happen. So, but nobody knows if they're, nobody knows if evolution has evolved it so that it can oscillate. It could be that it's tried to create a homeostatic system and has gone as far as it can go before it all oscillates, right? To try to maximize the homeostasis. But then humans come in and mess with it and just tip it over, and it starts to oscillate. So, and there's a bunch of synthetic ones if you're interested in those, and that's it. So what we're going to do now is we're going to do a little project actually, and a different kind of feedback. This has gained popularity in the last 10, 20 years. Looks like it's quite an important kind of feedback. It's called integral feedback. So what we're going to do is I'm going to give you a project to do. Might take you, I don't know, 15, 20 minutes. I don't know how long. But we're going to build this network, okay? So if you look at this network, so this is a different kind. Although it has negative feedback, it operates, the net effect is an operation that's quite different from the previous two we've looked at. And it gives it a unique kind of behavior. So what we've got are 4 steps, V1, V2. Do I have a better diagram here? Yeah, I call that V3 and V4. This is called V3. This is called V4, okay? Now what we're going to do is we're going to build this in stages, okay? So we're going to build the first stage without negative feedback, and we look at that. Then you're going to build stage 2, where you're going to add the feedback and then look at that, okay? Oh, sorry, so this is phase 1. So I want you to spend some time, maybe 10 minutes, building this model, okay? So this model has 2 steps here, V1 and V2, and 2 steps down here, V3, V4. These are the rate laws for the different steps. V1 is just mass action, V2 is just mass action, but V3 is mass action, but V4 is slightly different. It's a constant rate, okay? It's called 0th order because it's independent of the level of P, right? I'm going to call P the product. And I've got these funny little symbols, this funny symbol here. That just means we don't care what it is. You can put something there if you want, but if you do, make sure it's fixed, okay? Otherwise, just leave it out, all right? This X must be fixed as well. And you can use the following values for the rate constants, right? Then I want you to run a simulation from 0 to 100 time units and add the following event to the model. What this event will do is it will perturb V1, okay? All right. So, so there's a couple of questions here. I've been ignoring them, sorry. Let's see.
I think we've generally been
Yeah,
answering them
answer
as they
them. Okay,
go.
yeah. Okay, good. Yeah, the papers, there's a bunch of papers there you can look at, and they describe the oscillators. Not many papers describe how the oscillators work, but they will show you how they built them and so on. Is the yeast oscillation in metabolism not found in other organisms? It's a good question. I can't recall oscillations being found in non-yeast eukaryotes, in metabolism, but that might be simply because people haven't looked. There's a very, very strong feedback in glycolysis from ATP back to phosphofructokinase. It's very strong. Glycolysis is the main pipe for carrying flux into the cell, carrying material into the cell. It's very heavily controlled. And I wouldn't. And this is why it has the potential to oscillate. And the way you get yeast to oscillate is it's a funny. I mean, it's not a natural way. This doesn't happen really in nature. What you do is you starve it, right? You first starve the yeast cells, and then you add cyanide. Now the cyanide will kill the oxidative respiration, but will leave glycolysis intact. Now what you do is you give it a load of glucose. And if you do that, if you don't, if you give it too much glucose, the yeast will just die actually. But if you give it not too much glucose, the yeast will spontaneously start to oscillate in glycolysis. So it's a rather unusual situation. So it may not be natural. Nobody has come up with reasons why it would be good for yeast to oscillate like this. So it could be just a side effect of the negative feedback. Yeah, rat. Yeah, I I know there's been some work on looking at the transcriptome, and there does appear to be some periodic behavior in the transcriptome that's pretty wide. That involves, hundreds and hundreds of genes. So I think it's a case that people haven't looked much, and I suspect there's quite a lot of oscillation going on in cells for one reason or another. So any questions? So I'm hopefully you're trying to build this model. See what you get. Okay. So let's see if you get the model done by this phase one by because this is the biggest bit, by say 25 to the hour. So that's another 7 minutes. See if you can get this built in the next 7 minutes. If you have any questions about the model, just put it into the chat, okay? And if you do finish, I mean, if everybody finishes on time, ahead of time, just put up um put a hand up, all right, or something to say you're done. Um, I see the hands. Yeah, I can see all the participants. So if about, 67, 70% finishes phase two before 25 two, put your hand up, we can then move on to phase two. So if you finish phase one early, at least 60% of you do that, or 70% of you do that, we can move to phase two. Because I know not everybody will be doing this. So is this what we expect to get? Let's have a look. Yes, I think that's pretty much what you'd expect. Um, yeah. So the orange is P, right? Yeah, that's pretty much it. Yeah. How do we get out of this? Ah, sorry about that. Yeah. Everybody ignore that diagram, okay? That picture I just showed. Yes, X is fixed, all right? So either put a dollar in front of it or call it an external, okay? Initial value 10. Yes, that's right. Yeah. Yeah, my one looks slightly different, but it should be this, it's about the same though. I got. Yeah. Okay, good. We got a couple of people finishing. Thanks. I mean, once you get used to this syntax and Python in general, you can put together models really fast with this system. So the nice thing is you can cut and paste stuff and move things around and I mean other platforms have things like spreadsheets as the input mechanism. In fact, quite a few platforms do that. It doesn't work for me. It's too fiddly. So, but everybody has their own, preferences. Good. We got a few more. Okay, 3 minutes. Then I'll show you what I got, and then we'll move to phase two. Phase two should be easier. I mean, while we're waiting, I can mention that when we were exploring an idea whether we can get, AI to generate these models because they're just text. But we think it'll require some extra training. But in principle, they could help you build the models. You just got to be careful what you get, though. Yes to both. Oh yeah, sorry. Yeah, there you go. I mean, you can't ignore AI, so it's here. But at the same time, I don't want AI to turn into this black box and we end up with nothing. So if we can try to marry it with, mechanistic modeling so that we get explainable AI, essentially, I think that would be a nice compromise. I know people are rushing in to use AI for all sorts of things and using it in mission critical situations, I think is risky because you don't really know what the AI is doing. So um, okay, another minute or two, and we'll carry on. So this little model here is based on a paper from 2002 by John Doyle. Let me see if I can find it. I'll put it up if I haven't put it up afterwards. Yeah, I got the reference. I'll put that reference up afterwards. Okay. All right. I don't think we're going to get any more. Hopefully, most of you had a good chance at this. So let me switch to. Let me see. I must. Oh, don't I have them? I thought I had the model somewhere. Okay, hold on. Where did I put the model? Damn. I had the model somewhere. Oh, here it is. This is the model. Okay, so I happen to do it on Spider. I can bring it. Into. I can bring it into the this thing here. Let's do a new code. Let's do it now. I'm going to get rid of this because that's phase two. Okay, so this is the phase one model. All right. So 4 steps. So particular rate laws, I said. I have the event, and these are the initialization. So let's run this and see what we get. There you go. So for some reason, it's slightly different from one you posted, but it's basically the same thing. Oh, I know why, because I went up to 300, wasn't it? You went to, yeah, I think that's probably why. Or you went to, I see what is going on. Yeah, you went up to 100 and this should be at 50. I wasn't even listening to my own instructions. There you go. There you go. That's basically what you got as well. So we're applying a disturbance to K1, which is the first step, right? And obviously the system is changing. There's no resistance to this disturbance at all, which makes sense because there's no negative feedback anywhere. But the other thing to note is this P, the orange. The orange is basically is going to climb actually forever. It's actually going to go to infinity. It's going to go to infinity because it's what we call a zero order step. The rate is not dependent on the reactant, right? It's basically a fixed rate here that's leaving. And if the production of P here exceeds this fixed rate, P will just climb forever and ever and ever. All right. So this isn't a great model anyway, from that sense anyway. But let's see now. Why don't you go to the next phase and let's see what happens. So that's the thing we got. Let's see now what happens. So I said, what final value for E did you get? Well, it changed, right? It went up. It went up by a certain amount. All right. So, and then it stayed up. Okay. Right. Phase 2. So now add negative feedback, and that all that means is changing V1. And this is the negative feedback I've done. All I've added is a denominator, 1 plus P divided by Ki. The reason I add the 1 is so there's no chance of this going 0 and then getting a divide by 0. And there's no Hill coefficient on this. It's not a particularly strong feedback. In fact, it's quite weak. I do have a little constant here, which lets me sort of adjust some level of strength, Ki. So I'm setting Ki to 0.1, all right? And so that then adds this negative feedback to V1, all right? So add that change and then run the same simulation. Do the same thing as you did before. Run the same simulation, you can just play around with this. This is a very, very robust model. You'll see the effect. Should be pretty obvious, I hope, when, what you'll see, okay? Yes, somebody asked, in the example I showed, yeah, I already had the denominator in there, but and I commented it out. If you look here, yeah, that's potentially confusing. If you look here, I did have it. I did have that term in there, but this hash here means I commented it out. Because I wanted to show you phase 1, so I commented it out. So it's easy now for me to just uncomment it, and this is one of the advantages of text-based modeling, right? You can very easy to mess around with it. X equals 6. Did I put
10.
Oh yeah, I did put x equals 6. That it should be 10, right? So let's do that. And then yeah, you should get it doesn't matter if it's not exactly like that. The two key observations is that yeah, E isn't resistant at all to that perturbation. It changes. Went from 5 to 10, right? I doubled K1 and it doubled as well. That's really not very resistant. But the other thing to note is that P just keeps on climbing. In fact, let me show you. If I go to 1,000, right? P just keeps on climbing. In fact, it'll never stop. So that's not a great thing, right?
It's like the zombies.
Yes, a bit
The
like the
other one.
zombies. We don't want to live in a zombie world. Okay, so hopefully most of you have done that change, I hope. So okay, so people are asking about the event. Yeah, let's find out what a good set of conditions are. So I've uncommented that.
You meant instead of 3 time units, it was 300 time units.
Well, it could be, yeah.
And
Maybe that would
that's
make
what you
sense.
had, right?
That's what makes sense. 300 time
Yeah.
units, yeah. And I probably put that as 100
that's why that was 100 too,
just
right?
to
Yeah.
see a nice delay, I guess. Okay, let's run it. Hopefully you see. Oh yeah, I remember fiddling with this. Hopefully you'll see something like this. As you can tell, this is quite different from phase one. First of all, we can see that although E responds initially, it gets pushed back down by the negative feedback. The other thing you notice is that P doesn't go off to infinity anymore. It actually stabilizes at a higher value. What's interesting to look at is what I'm going to do is, let's see, what's the best way to do this? Yeah, okay. I'm going to take out this event, right? Because I want to record the value of E, okay? So this is the normal steady state level of E. It's actually 0.6, okay? All right. That's without any perturbations, right? Okay, so now let me add the perturbation now. Run it. And let's look at E again. Now this time at about time 300. And you'll notice it's 0.60548. That looks pretty close to the original. In fact, let me extend this all the way to 3000. And let's print that out again. 0.5999999. In fact, E is being restored exactly back to where it was. There is not an atom's width difference between E before and after the perturbation. This is called integral control or exact adaptation, or no, perfect adaptation, right? The system adapts perfectly to the perturbation, i.e., it doesn't shift from the original. And this is a property of what's called integral control, right? And in order to explain this, I need to go back to the last thing we'll do, okay? Where are we? Here. Need to go back to this diagram here, explain what's going on. It's not difficult to understand what's going on. What's happening is, remember that before we added the feedback, this P was increasing forever, all right? Once we add the feedback, and I make the disturbance, P will rise, all right? But what's happening is, as P rises, it's pushing down V1 more and more and more because it's acting as a repressor, right? And it'll keep on pushing down V1 until E is restored back to what it was before, because it's only at that point that P stops rising. All right? So I'll say that again. So if I make a disturbance in V1, E goes up, P goes up. Now, because of this zeroth order, P will continue to rise. But as P rises, it starts to squeeze down V1 more and more and more. And as it squeezes down V1, E starts to drop. And once E reaches the original value it was, P stops rising. And at that point, it's reached its new steady state. And the reason why it's called integral control is because P is considered the integral of E, as in calculus, right? It's considered the P is essentially integrating what E is doing. So if E returns back to where it was before, essentially P stops integrating. Whereas if E is slightly off, P will integrate that difference, right? And originally you saw that P was a straight line. It's a straight line because it's integrating a constant level of E. Remember, an integral of a constant is a straight line, okay? So it's actually an integration. And that's why this works, okay? So we've come to the end of that, 10:45. Are there any questions before we have the break, and then John Gennari can take over? Yeah, somebody asked, yeah, let me get you the model. Yeah, I'll cut and paste into the chat, okay? So there it is. Very robust, this model. It's very easy to get it to work. You're wondering, well, does this get used at all in nature? It does. It's used in some very clever ways. Most famous is E. coli sensing. So the way E. coli senses nutrient changes is through an integral feedback controller, and the the way it uses the integral controller is even cleverer than this. What it uses it to do is to reposition the response of the receptors on the surface of E. coli. So E. coli at its front, so to speak, has all these receptors for sensing nutrients, all right? Now, the receptors can only measure the changes in the nutrient over a narrow range. So what the integral controller is used for is to move that threshold up and down, which means that E. coli can detect a very wide range of inputs. And that's what the E. coli uses there. And you're wondering, well, what's the integrator? The integrator is actually the methylation of the receptor proteins. So as it methylates the receptor, that's the actual integration itself. Now, it's not a very wide integral range because there's only four methylation sites, but it's enough to make the whole thing work. The slides, yeah, that's fine.
Yeah, do you
Oh,
want to
sorry, guys.
rename this thing from Untitled Zero to?
I sent a direct message again. Hold on for a second. I
Oh,
keep forgetting to change the.
to change it to everyone.
So there's the model to everyone. So what's the question is in?
Just give this notebook a name besides
Oh
Untitled
yeah,
Zero.
yeah.
Yeah.
Let's call it day 3, shall we? No, call it day 3
It's day, and this is
plus.
the same one you used yesterday, so day 2 and 3, I think.
Oh, okay. We'll call it day 2, 3 then. Oh, can't type. Yeah. Day 3. Okay, that's what it's called. Day 2, 3. All right. Okay. So before you all go, we've in the past, people have mentioned it'd be nice to have, shorter shorter time online schools for on specific topics. That's certainly an option. We could do something in winter or next spring. If there's any interest in specific topics, that take an hour or two, we could certainly do that. We could cover more advanced topics, or we could cover stuff that we haven't covered at all. I'm sorry, excuse me. So let us know if that is of interest. Maybe we can put out a poll or something. Oh, sure, excuse me. So let's have a break. Let's come back at roughly 11, and then John Jerry can take over. Okay.